Thank you, Chenglu. Yeah, it's my pleasure to introduce today's seminar speaker, Dr. Kent Daane. And Kent did his bachelor's in zoology at the University of California in Santa Barbara. He went on to do a PhD in entomology at UC Berkeley. And he's currently adjunct faculty in the Department of Plant Science at California State University in Fresno, California. He has held that position since 2007, and since 2006, he has been the cooperative extension specialist in the Department of Environmental Science, Policy, and Environment at the University of California, Berkeley. Kent, his research projects are on developing IPM programs in California's vineyard systems and tree crops. And he's working on the discovery, importation, and evaluation of 3 Asian native larval hymenopteran parasitoids for introduction against the spotted wing Drosophila susukii. And that's, I believe, what he's going to talk about today. All the work that he has done in foreign exploration and hopefully to release these parasitoids to control this problematic pest in fruit crops. Kent received the Distinguished Scientist Award from the International Organization for Biological Control, the Nearctic Regional Section, while I was president last year, and he gave an awesome talk at the ESA. So after his talk, I was very encouraged to invite him to give this seminar for our department. So thank you, Kent.
Great, thank you. So what I'll do is I'll start by sharing my screen. This up. Good. Does that look fine for everybody? So what I'm going to do today is to kind of, I'll get to the spotted wing Drosophila story a little bit later. What I want to do, though, is to kind of take you down this road of how biological control, classic biological control, has changed over the years. And I don't want to date myself, but I've been a part of that change. I've seen the different philosophies go from releasing almost everything that you think might be potential to being very cautious about what you release. And in fact, this famous picture from the California Biocontrol Group, taken in 1948, I'm going to make a connection to that picture later on in the presentation. So for that reason, I'm actually going to have a long introduction before I get to spotted wing Drosophila biocontrol by talking about the first biocontrol project I ever worked on. And that will get us into the right frame of mind to open up some discussion at the end of this about what is the proper way to import, evaluate, and release a natural enemy. So everyone knows pest population graphics, economic injury level, trying to keep pest populations low before the crop gets to a critical point where it can cause damage. With spotted wing Drosophila, that's typically when the fruit are ripening up and the pest can actually oviposit into the susceptible fruit. So most common thing is to apply an insecticide to drop the population down. And you have to oftentimes repeatedly apply insecticides for spotted wing Drosophila throughout the season because you're going after the adult stage. The hope in classic biocontrol, not augmentative biocontrol, but it's the introduction of an exotic natural enemy, typically from the area of origin of the pest, in this case, Asia, for the spotted wing Drosophila. So you look for, find, and release a selected natural enemy. And the hope is that you get this oscillating pattern of movement below the economic injury level. And the hope is that this is sustainable over long, long periods of time, only broken typically if there's an insecticide application that disrupts the natural enemy. So what I want to do is not talk so much about this process after the release of the natural enemy, but I want to talk about what's going on before we select and release the natural enemy. That's that pre-selection or the quarantine work and the changes that have happened in my career on how this process develops and how you do a successful program. So I'm going to do this going back to my title slide of Changing Attitudes and Policies in Classic Biocontrol. I'm going to look at this change from natural enemy impact focus to risk aversion focus. And I'm going to do this in part with this long introduction, an introduction that starts with my first biocontrol project, which was with the black scale as a pest of olives. It's also a pest of citrus. And then we'll get to more of the data set when we talk about spotted wing Drosophila and Ganaspis brasiliensis, which is the picture below. Up above, that's black scale with a parasite attacking it. And so this long introduction will be more of a story, less data, but just kind of a story to tell you how things used to be done, where my mind frame was, and how I got to accepting risk aversion as a better avenue to practice classic biocontrol through. So remember that picture up above looks like a lot of guys from the 1920s, was actually taken in 1940s. These are folks that really started biological control moving from something being done by the State Department, what is now the USDA, but it used to be done by the government, and it was moved by this group really into the universities, and now it's working hand in hand with the state again. Harry Scott Smith is circled in blue there, Prof. Smith, because he actually did not have a PhD, but he was in charge of the state of California's biocontrol program. And when it moved to the University of California at that time, UC Berkeley was really the only campus doing agriculture, and UC Riverside was just an experiment station, as was UC Davis. So Prof. Smith was in charge of the UC Riverside experiment station, which at that time, Davis and Riverside were experiment stations of Berkeley. So he actually was a professor at UC Berkeley initially. Before him, after the Vidalia beetle success, Howard and Fisk and others had started to think about, well, how do we harness this biocontrol program? Because immediately after the Vidalia beetle, there was a period, and that's a beetle that was released in citrus to control the cottony cushion scale, an amazing success story. There was this period in North America that they called the lady beetle crazy period. Folks were going all over the world, and if it were a lady beetle, they would import it and release it, not even knowing what it attacked. Just the idea that anything that's a lady beetle might have the same success as the Vidalia beetle. And so Howard and Fisk started to discuss what really a parasitoid and a predator were important to have, what qualities they were important to have. Smith described dispersal, rate of population increase, and searching ability. Well, two of my mentors were Ken Hagen and Carl Huffaker. I actually grew up with Carl's son. I grew up in high school, sneaking beers out of their party and listening to stories from Carl and Ken and Vandenbosch and others arguing about this idea of biocontrol maybe needed to become more of a science and less of an art form. And so DeBach and Schlinger, who I also met and knew, started talking about what are good, and in that picture in the bottom is just me, and you've got Daane Gonzalez Walker Jones, Joe Coleman, and there's Ken Hagan, who is up circled yellow in the upper picture. Down below, this is in Hermosillo in a meeting down there. So Debach and Schillinger started talking about attributes of natural enemies. They included things like adult female mobility, adult female longevity and fecundity, oviposition behaviors, oviposition in suitable host species, not a specific host species, but in suitable host species. Sex ratio, we wanted to have more, a female biased sex ratio was always considered to be important. And of course, along with that, development times with respect to the host. They always thought you wanted to have a faster development time than the host, so you would have multiple generations per individual host generation. And of course, persistence in the host area, the host habitat. So with that in mind, let's look at how their teachings to me were brought into my first biocontrol project. And that was working on black scale on olives in California. We see on the left the damage that the scale will do to the olive tree. It's a pest of many, many crops, but specifically olives and citrus tend to be two of the more important crops. And I found out meeting with Cesar's students today that Pablo works with Alejandro Tenya in Valencia, Spain, who worked on black scale and still works on black scale. So I put in a picture of Daane Gonzalez, who you saw in the previous picture, also one of my mentors, but he's part of this story as well. So I put some photos of him in. So what we're seeing here are 4 of the most important species. And what I just showed on the left is a list of all of the different parasitoids that have been released in California against black scale. And you can see this goes from a period right after the Vidalia beetle was released. So the Vidalia beetle in citrus led to this other citrus olive project where they were going out and getting natural enemies from all over the world: Australia, South Africa, West Indies, Hawaii, China, Pakistan, Australia. In dark black, these are the parasitoids that we know have established in California. In light gray, these are all the species that were released and not established. And being established doesn't actually mean that these insects were adding to control. It just means that they were found years afterwards. If we want to see what adds to control, the 4 species that you see here at the time, called Metaphicus bartletti, Metaphicus helvelis, and Metaphicus lounsburyi, and this Scutolista, which is a pteromalid, those are the ones that are commonly found and are really contributing to some level of control of this. And we've also got these just advent parasitoids, all Cacophagus species, all facultatively hyperparasitic. So Cacophagus, Lysimnia, Scutolaris, and Crassius can all attack black scale, but if there's a parasitoid in black scale, a Metaphicus species, for example, they can switch over to being a hyperparasite, and no one's claiming responsibility for releasing these, but they probably were brought in with some of these early releases because they've been known to be in California since the 30s, 40s, and 50s. So what was my role in all of this? Well, if we look down here at the very last parasitoid ever imported and released in terms of classic biocontrol for black scale, that was my thesis project. And when I came into this, the importation process was not what it was like today. The screening process was pretty much a simple check to make sure it's not a hyper, to call up the USDA, get permission and do the release. So in this case, we were contacted by a colleague of my major professor in Spain, and they said that there's this species Metaphicus zebratus, which is also attacking black scale, and which I don't think you've got in California. And so Leopoldo Caltagirone said, sure, send it to us. They sent it to us. I was asked if I wanted to work on this as part of my thesis work, because I was looking at parasitoids in olives around California. And one of my mentors, Chuck Kennett, who worked with Carl Huffaker, was retiring. So he didn't want to do the release. And so I just kind of included this. And then with Leo, we started to say, I don't even know if this is zebratus. I was learning how to be a taxonomist. It looked different than Lounsbury, certainly, but it was fairly similar. But it didn't look like zebratus was supposed to look in the Keys. And so as part of my thesis, I also started looking at the taxonomy of this. So the important thing to note is that we got this material, we put it in quarantine. We said it is Metaphicus. We're not sure it's actually zebratus, but it's Metaphicus, and all Metaphicus are not hyperparasites. So called up USDA, and within a week, it was out of quarantine and in the insectary in colony. We wrote to the California Olive Committee, and we got funding for release, not for importation, screening, or post-release, just to release this natural enemy. So I put this out. I started to make more collections, and one of the first things I found was other species not known to be in California, this Cacophagous rusti and a Procacophagous probus, that had been released, one in the 1930s and the other in the 1970s. And again, this emphasized that it was not important to do post-release surveys. You put the material out there, and if it takes, it takes. If it doesn't take, you kind of move on with it. Another thing, remember I mentioned that we didn't feel like this was zebratus. Well, during my thesis, I started communicating with John Noyes at the London Museum. And I said, I don't think that this is Metaphicus zebratus. And sure enough, he was looking into this, and he said, well, actually, Metaphicus bartletti type specimen is, in fact, Metaphicus lounsburyi, named earlier, which meant that Metaphicus lounsburyi couldn't be lounsburyi. It had to get a new name, so it was Metaphicus annechi. The species that we released, I named after Ken Hagen, so it was Metaphicus hageni. And later on, the pteromalid was also renamed. So not long after my thesis work was published, all of the insects in the papers were actually different species, just emphasizing the importance of taxonomy. And it also becomes important today. So were we just not understanding what was going on? I want to emphasize that my thesis committee, Carl Huffaker, Chuck Kennett, one of my mentors, Leo Caltagirone, Ken Hagen, Don Dalston, these were huge people in classic biocontrol. They all were hugely successful, and part of understanding and pushing biocontrol from an art into the science. And in fact, Carl Huffaker had thought to great extent the question of screening and selecting natural enemies. And in fact, he and Chuck put out a paper discussing this very same thing, where he said that you really cannot tell which natural enemy is going to be the best natural enemy. And so it's very difficult to release only one species and know you're releasing the right species. And part of this was because in the facilities we've got available, meaning the quarantine, you can't possibly do the kinds of studies that you need to understand what's going to happen in the field environment. And at the origin, it's going to have a different environment, meaning where you collect the natural enemy, than in the release habitat where you're releasing the natural enemy. So while this is all going on, while I'm a graduate student, work is coming out, led in part by Howard in Hawaii, about the environmental impacts of plastic biocontrol. So I used to be considered to be someone looking for alternatives to organophosphates, someone going towards organic research, sustainable farming. And all of a sudden, people were saying, well, maybe classic biocontrol is a bad thing. And in fact, with Daane Gonzalez, who was a professor of Riverside, my next two classic biocontrol projects were soon after I was hired. And it looks on the resume like I've switched jobs a lot. It's just that UC Berkeley keeps on changing the name of the department. So I've been with Berkeley for about 32 years now. And two of the initial projects were with Daane Gonzalez and with Sergei Trapitsyn on the obscure millibug. We started this argument about how important is taxonomy and how important is understanding the host specificity of what we're bringing in and what we're releasing. And Daane was adamant that it doesn't matter. You just release it and the good ones survive and the bad ones disappear. And Sergei and I were adamant that no, there's got to be more of a screening process that goes on so we know what we're releasing. And in fact, Sergei, later, these parasites we released against leafhoppers, they're all an Anagris group species. He ended up renaming most of them because I had saved them all, where Daane called them Anagris epos. We found out they were in fact a collection of 4 species. So this is going on at the same time that a lot of people are now starting to question screening process and classic biocontrol. And if you break this down, this is really nice work by Matthew that came out in Biocontrol in 2016. You can see what I consider to be this Vidalia beetle crazy period from the 1870s to the 1920s, a drop during World War II. And then it picked up with my mentors, the Ken Hagen, the Carl Huffakers, the post-war group that really introduced me to biocontrol, really increasing the number of natural enemies being imported and released. And then this period that happened from the beginning of my thesis to today, where there's been a shift from impact to risk aversion. And you can see this in the graph over here, where we've got a reduction in the number of introductions, but an increase in the percent established. And where my insect kind of fell into was this group that established, but did no harm, but really did not do that much to control black scale. So perhaps we could have done better with the screening and the quarantine, but it did no harm. So, and this is really nice work by George and Matt that kind of summarizes this shift of an era from accepting more risk because we're looking at impact to now today accepting less risk. And we can see that in the processes that I have to go through to get an insect out of quarantine. So I have to file a petition that triggers compliance with 2 processes, an Endangered Species Act and a National Environmental Policy Act, both of which we do address risk. The petition has to have 7 components to it. The proposed action, it's a pest, why we want to release the natural enemy, the targeted pest information, biocontrol agent information. This oftentimes takes 2 or 3 years in quarantine. Host specificity has become the primary concern. And then what's going to happen post-release, pre-release clearance, post-release monitoring, and then it goes through a series of bureaucratic and scientific reviews. The NAPO process is really the major scientific review. These are all bureaucratic processes, including going through tribal consultation, publication in the Federal Registry, and then back to APHIS to develop a FONSI, final publishing of the FONSI permission, and then permission to release. So I told you that introduction was going to be long. It was longer than I expected, but let's see how this has changed attitude towards biocontrol with classic biocontrol of spotted wing Drosophila. This has been a group effort with many people involved. And I'm sure once we get this parasite, many of you folks at Rutgers will be involved with the release as well. First thing you do in any biocontrol project is you look to see, are there natural enemies here in North America that we can manipulate? We found that there are two pupal parasitoids, Pachycropoideus and Trichoprea, no or very, very rare larval parasitoids. We're looking at augmentation, but basically these natural enemies don't give control of spotted wing. So next phase is to go to the origin of the pest. We were lucky in that we knew where spotted wing Drosophila was originally from. We focused on southern China, collecting in areas around Kunming and throughout South Korea. And I want to thank all the collaborators. Whenever you do this, it's key to get collaborators in the area of origin. So we worked with wonderful people in China. We can see Kim Homer from the USDA as part of this. Scientists in Cabby, Switzerland, based in Italy, were part of this. We see Massimo and Guillermo from Italy that are primarily taxonomists, but help with collections. And a special thanks to Young Soo from South Korea and Nancy and Shi from China. And a special thanks to Xinqing Wang, who's out your way now in Delaware, USDA. The middle picture and the lower middle, who really did a tremendous amount of work on this project. So now what I'm going to do is I'm going to connect the young Kent to the, I'm going to say the current Kent rather than the old Kent. I'm going to connect what people used to do in biocontrol to what I'm doing today and try to understand if I'm doing the right thing. So these are all the parasitoids we reared out of those collections from China and South Korea. Altogether, 19 parasitoid species were found. Well, if this were the 1920s to the 1950s, I could make an argument to release all of these insects by saying we don't know what's going to be important, not important. And what I've got here in this color code is to show you that the 5 most common insects we reared were the 2 pupal parasitoids, Pachycropoideus and Trichoprea, a braconid, Asobara japonica, and these 2 phagidids, Leptopollina and Ganaspis, Leptopollina japonica and Ganaspis brasiliensis. Those kind of came out as the 5 most common species. If we looked at what we knew from our collections, we're being reared primarily or exclusively from spotted wing Drosophila, we would come up with these 8 parasitoid species. And those included a number of braconids, all Asobara species, and the other 4 we talked about, Ganaspis, Leptopollina, basically the phagidids, pteromalids, and the diapriid. So then I can make an argument for maybe releasing these 8 species. The 3 less common Asobara, you might argue that they were coming out of spotted wing or closely related species, and maybe they're not common in China or South Korea, but they would do fantastic being released in Washington, whereas maybe Ganaspis might do better in Oregon or California. So still the 1960s to 1980s biocontrol practitioners could kind of make an argument for these 8 species. And in fact, I think they would. But if we look at which of these things really had an impact, we're being reared out of Drosophila suzuki'i. Most commonly, it comes down to the braconid and the 2 phagidid species. And we could also get some idea of what was going on from these field collections, which becomes more important today, rather than just that first project I work on. Remember, I never went to Spain to get this insect that was called Metaphycus zebratus. It was just a colleague of Leo's in Spain in a conversation saying, hey, we've got this thing that we're working on. Do you want it? So we had no idea of its biology in the field, whereas on every single one of our collecting trips and with our collaborators, we were collecting data on these parasitoids in South Korea and China. And from this, we were getting an idea of the biology of these three species that we felt were the most important. So for example, we knew that the Asubara was always collected later in the season, and we knew that we were getting it and rearing it from Drosophila melanogaster later in the season, as well as Drosophila suzukii. And we knew that the two Phagidids, Ganaspis and Leptopolina, were more common earlier in the season, were only being reared out of Suzukii, and were getting a little bit pushed out later in the season by Asubara japonica and the other Asubara species that were less common. We also could see some early things. These are data out of China in 2016. So for example, in the top graph, and these collections that I'm focusing on right now, what's interesting to me about this particular collection is that we wanted to go this year. We were collecting between 2013 and 2017. In 2016, we were going early in the season. So the 2013 collections were in July and August. Then we went in July. Then we went in June and July. And in this one, we went in June. And so we were collecting on mountaintops where at times there were still patches of snow. We were collecting from different Suzukii host species. We can see here it was two different rubus species, Fragraria and a Sambucus. Sambucus is elderberry, basically an elderberry-like species. And the Fragraria is just this little mountain wild strawberry. And so these represented different types of host plants. The Sambucus was a large bush that could grow up to 15, 20 meters in height. The rubus were like wild berries. The Fragraria was growing no more than 15 to 20 centimeters off the ground. It grows like a little mat on the ground. So in the top graph, looking at the parasitoid complex, what was interesting to us is that the Asubara early in the season, that would be the little blank white group coming off of the rubus. I can do this with this. So this would be the Asubara group. This would be the Leptopolina, and this would be the Ganaspis. So we were getting Ganaspis and Leptopolina at these different collection sites coexisting with each other. Ganaspis was a little bit more common. And in terms of parasitism level, so this is just parasitoid complex. Here's percent parasitism rate, and there's all kinds of arguments you can make for the good and bad of percent parasitism. But we saw about 20% parasitism in most of these host plants, but up to 60% on the Sambucus. And our highest parasitism levels were 78%. So these two species look pretty good. We brought them back to quarantine. And I want to point out the weaknesses that Carl Huffaker and Kenneth talked about doing work in quarantine. And clearly, I mean, we're doing studies with Suzukii, the fly, in these little Drosophilid vials. So this has nothing to do with reality of what we're going to see in the field, but it's what we have to work with in quarantine. We looked at all kinds of biological parameters. I'm going to pick out a few just to show you. Looked at interspecific competition and looked at non-target impacts. So biological parameters were just basic biologies. So for example, female survival and egg production as an every day or every other day. Basically, Ganaspis produced about 98 offspring per female, Leptopolina about 107. So very close to each other, lived for about 17 to 18 days. Asubara japonica in that same range, about 117 offspring per female, about a 16-day adult period when provided host. So very, very similar to each other. The two Phagitids both preferred to oviposit in smaller hosts. So this is host age of the fly, 1 day, 2 day, 3 day, and 4 day. Then they start to go into pupa at these conditions. And so we can see here that both Ganaspis in white, Leptopolina in gray had much greater parasitism levels. And these are host parasitism after a 24-hour exposure period, just to make sure that's clearly understood. So both were about the same. They preferred 1 day and 2 day old hosts. And there was a significant drop off in 3 and 4 day old hosts, but they could still attack them. Asobara, in contrast, we look here again at host larval age, 24-hour exposure period. It really didn't matter to Asobara how old the host was. A little bit of drop off here as the host larva is preparing to go into pupation. A development time, it took a little bit longer to develop, a little bit shorter to develop. I mean longer if the host was smaller, shorter if the host was older. We see that with all kinds of parasitoids. There's a drop off with the older hosts because the parasite has a shorter period of time before the host pupates. And that has an impact on host vigor because you get a reduction in host size in older hosts because they've got a shorter development time. And this smaller host leads to a smaller parasitoid, leads to lower fecundity. And we found that across the board with all 3 of these species. We look at functional response, that's the number of hosts attacked across host density. And we can see that they all have a type 1 linear functional response. I just want to point out that Asobara, much more aggressive than these. You can tell that with the slope of the line. So Asobara, a little bit more of a monster in terms of attacking in quarantine conditions. And in fact, we look at interspecific competition, and we see that Asobara and Leptopolina develop a little bit faster than Ganaspis. And another basic rule of parasitoids is that if you develop faster from the egg to the larval stage, if you're the parasite racing to the first instar, that first instar is typically then going to beat out another egg of another parasitoid that develops slower. And in fact, when we look at competition, what we've done here is we're giving them 5, 15, or 30 hosts per 24-hour period, looking at production, and we're seeing that there's an increase functional response with an increased host density, as you would expect. So let's just look at the higher density. If we release only Asobara, it produces more offspring than when we release only Leptopolina or only Ganaspis. When we release Asobara and Leptopolina, altogether there's no increase in total parasitism, but Asobara and Leptopolina kill about the same number. When we release Leptopolina and Ganaspis, Leptopolina again produces about the same number as by itself or with Asobara, but Ganaspis goes down dramatically. And in fact, we found that Asobara and Leptopolina that develop faster don't care if they're ovipositing in a host that's already been parasitized by something else. Whereas Ganaspis avoids hosts previously parasitized by Asobara or Leptopolina. So in thinking about what to release, the first parasitoid we would want to release would be Ganaspis to give it a chance to establish in the North America before we look at Leptopollina, knowing that there's this competition. But the most important thing we do in quarantine is looking at non-target impacts. Non-target impacts is that risk aversion. So you take a natural enemy, ladybird beetles, you release it against aphids, it kills the aphids, that's great. But the non-target is, let's say that lady beetle is a generalist and it starts feeding on larvae of an endangered butterfly, that's bad. Or it's got some biological trait like the Asian lady beetle that is a pestiferous. The Asian lady beetle overwinters in people's homes, in their attics, and on their windowsills because it became kind of a pest. And for me, this really hit home with the Southwest willow catcher that was part of the Tamarix biocontrol project with the Tamarix leaf beetle. The biocontrol project is working, getting rid of Tamarix trees, but those trees were becoming a host for this endangered Southwest willow flycatcher. And this really changed the USDA's attitude on giving permits. And we went from impact to risk aversion. So in quarantine, we look at that. We can do things like looking at attraction using an olfactometer from these natural enemies to different fruits. And we found, for example, in the first two, that all of these parasitoids were more attracted to rotted fruit or infested fruit than to clean fruit. Shown here again, more attracted to infested cherries than uninfested cherries. But they were equally attracted to rotten cherries as well as infested cherries. So even with the olfactometer work, we thought, well, maybe some of these might have some non-target impacts because they're really just focused on any damage to the host itself. And it didn't matter if it was cherries or raspberries. So the most important thing becomes, what else can they attack? And this became our primary focus in quarantine. We looked at 24 non-target Drosophilid species. And this included things that we could not even rear on their native plants, so we had to rear everything on diet. What you see here, along here in a relationship phenogram, these are the different Drosophilids we tested. These are the offspring per female per 24-hour period for Asobara, Leptopollina, and Ganaspis. What becomes clear is that Ganaspis attacks this melanogaster group, melanogaster, simulans, and Suzukiae. We had this one attack over here, which might have been a mistake of us in quarantine, but we'll deal with that later in the discussion. Asobara pretty much got reared out of everything. Leptopollina really focused on the melanogaster group as well, but there were 1, 2, 3, 4, 5, 6, 7, 8 other species it did attack. And remember, this is in quarantine in these little Drosophilid vials. They either attack this or they die. I also want to point out something else. Here we see Ganaspis attacking spotted wing on top of a blueberry. Well, why our native Phagidids can't attack successfully Ganaspis is because spotted wing is because so many of these flies have this immune response. This is the egg of the parasitoid, and the fly can coat that egg with flattened hemolymph cells, and that melanizes the egg, and we can see it here. These are two parasitoid eggs that could not develop. So that's an immunity that some of these flies have against these parasitoids. So when we look at these different flies, and we look at how many of them had a black capsule in them, we can see not many in Asobara, and that's because almost everything successfully reared out. But we can see Leptopollina and Ganaspis were not discriminating against the host. Again, this is an artificial diet, They were trying to attack everything, but the host had an immune response against it, which is really good for biocontrol, because that means that even if they host shift in the environment, because there's not a lot of spotted wing and melanogaster, and they attack something else that like Willistonia here, it won't matter because that fly's got a defense against it. So what I want to do in the last 5 minutes is to bring in another complicating aspect of this. We know now a lot about these parasitoids, but we know maybe a little bit too much. We know that Ganaspis can be separated, and what we did here is just using the CO1 gene, we can separate these out into different strains or populations. And most of what we got in South Korea and China were part of this G1 population. And most of what we also got, about 20 to 30%, were part of this G3 population. And the G4s, G5s, G2s, this is just from a library bank of other Ganaspis brasiliensis, but we never reared these out of spotted wing or its closely related species. And in fact, our colleagues in CABI, working with the same complex, believe that these represent cryptic species, that this G1 population actually is specialized and will only attack spotted wing, whereas this Ganaspis brasiliensis will attack spotted wing and other species. And they showed this in one of their articles, just like our work, Leptopolina acebara attacked a larger range. They didn't look at as many host species, but they showed in this initial work that G1 only attacked Suzukii and only in fruit, not even in diet. They have since changed that and come over closer to our work that it also will attack it now, melanogaster in fruit and Suzukii in diet. So our two programs are very, very similar, but let's go back to my little what if Kent Young and Kent season from experience. So my gut feeling right now is that both G1 and G3 are so similar. For all intents and purposes, we're calling these the same species. We can use molecular work to tell them apart. But in fact, another study based out of USDA with Matt Buffington using a greater molecular description shows that you really have a difficult time separating G1 and G3 populations. So my gut feeling is that we should release both. And in fact, Kent Daane from 15 years ago would argue to release both Leptopolina and Ganaspis brasiliensis. So going back in time to kind of conclude where we are right now, I think that there was an argument from the 1920s to 1950s to release all of these insects. I can see the argument from the 1940s to the 1980s to include releasing those acebara species that were not very common, and in releasing the pupal parasitoids, the Pachycropoideus and the Trichopria. Even though they're here in North America, maybe these strains in China are a little bit better, and acebara, Leptopolina, and Japonica. I think that the more modern Kent philosophy would have been Leptopolina and Ganaspis, and you'd start with Ganaspis because of our quarantine work showing competition between these two. But even in the 1990s, I would have excluded acebara japonica because of its competition against Leptopolina, and because it was showing up later in the season, and because it had such a wide host range. But I want to point out that our group came up with Leptopolina and Ganaspis immediately based on the field work we did in China and South Korea. Getting it down to just Ganaspis brasiliensis to satisfy the new requirement going from impact to risk aversion took an additional 4 years of quarantine work and about $250,000 in research. And that's not the end of it, because we submitted, this is what we have to go through. So again, getting this petition together took an additional 4 years of work. Submitting the petition, it was submitted in 2018 and then again in 2019 because they wanted more evidence of the genetic separation of the two Ganaspis. Getting it from this point to where now it's waiting publication in Federal Registry has taken 1 year and 6 months. So remember, it took me 1 week to get Metaficus, what now is called Hageni, out of quarantine and being able to release it in the field. It's taken us from this point to where we are today about 5 years. And once it clears this hurdle, once it clears the FONSI, supposedly it goes within 3 weeks back to me. So to conclude on this, my attitude has changed. Part of it was, as a graduate student, you want those projects. You dream of, oh, I'm going to release this insect and tomorrow there's going to be a parade for me because I've controlled spotted wing Drosophila. And now I take the philosophy that biocontrol is not your project. It's this group project, because it might, whatever you release may not even have an impact until 10, 15, 20 years after you release it. You're releasing a tiny amount that has to readjust to a new environment and build up. So we've gone through this change, and I think it's still, though, a combination of art and science, and that's because of the limitations we've got in quarantine. You have to decipher what you're reading with what might happen in the field. And for that reason, I wish agencies would give us more money to give to people in the area of origin of these pests, so we can have folks in China and South Korea study in the native range what these natural enemies are doing. I also think from the current study and going back in history that we would just be so much better off if immediately on importation, we had taxonomy experts that can tell us exactly what we were collecting from the different areas, so we could know if we should do isolines immediately. That's how we lost Ganaspis G1, because we didn't keep the population separate. We moved to diet, and the G1 eventually was outcompeted, or we just lost it from the G3. And so I think the USDA should keep the vigor, maintain the rigor of what's going on, but speed the process up. I don't mind doing the non-target work, but it's been very painful waiting for this petition to clear permission through the bureaucratic process. And just the last 2 slides, what do we do now? Well, we've got these colonies going in California and at the USDA in Delaware. Our hope is to, once it's cleared from quarantine, to start sending material out, developing regional insectaries once it's out, and having them release this material. And I want to point out that Ganaspis is now in Canada, and Leptopolina is also in Canada and in Washington. So we're working with the USDA to be able to get this material based on our studies moved around the country as well. So with that, I'm going to thank you. And none of this could have been done without the help of so many postdocs and collaborators. And I want to thank all of them as well. So with that, I stop my share. And hopefully that was fun enough that people got a little bit of information and not just a bunch of graphs.
Thank you. That's excellent. Also, questions for Dr. Daane?
Well, may I ask a question real quick? I'm actually meeting with you at 3 o'clock, but
Yes.
I have a question about what drove, and I'm sorry if I missed it, but what actually was the driver for increased caution? Was it other researchers? It came from researchers? It came from farmers? It came from the public?
It started in North America, it started with the weed biocontrol folks. And so as early as the 1970s, they were meeting as a group and suggesting that the negative aspects of their mistakes would have greater ramifications because they were releasing oftentimes herbivores into the environment. And so that would have greater public concerns because it would be more aware publicly. It started with the entomologists. That's what was tied in with that Tamarask project. It started with the entomologists, really not just in North America, but on a worldwide basis. I think in Europe, they were getting a little bit concerned because some countries were more pro-classic biocontrol and other countries were not so much. And then we started to have problems with Howard's work in Hawaii in the 1990s. And then a number of excellent articles after that. I forget right now, his name is escaping me, but they talked about the Pandora's box. Basically, the idea came that once you release it into the environment, you cannot pull it back. It's out there. So that's really what started to change what was going on.
Right. So because I mean, I work with mosquitoes and you know, gene drives and transgenic mosquitoes is sort of the equivalent in some respects to what you're describing. And so it's fascinating. But I mean, at the same time, the alternative is insecticides. So
Yeah.
how did the insecticide, you know, how did that build in there?
So I know that I mentioned this earlier this morning. In traveling in Spain, talking with a researcher there about classic biocontrol, he mentioned that organophosphates were far safer than biocontrol because we knew exactly what they were going to do. We knew how long they were going to last. We knew after 2 months, we were not going to find this in the environment after we made the application. And yet, if you released a natural enemy, you don't know what's going to happen in 20 years. And of course, the biocontrol practitioners are banging their heads against the wall with what you're doing right now, because it's getting so impossible for us to get things out of quarantine, and 20 years ago, people would say, release a GMO? Oh, that will never happen. And now it is happening.
Mm-hmm.
And it's a different avenue that you have to go down, but it's the same agency. But they've got, in part because we did not write the rules correctly. We allowed the fear to write the rules for us. And then when the Tamarask problem came out, I've talked to folks in the USDA that said, weed biocontrol projects are scrutinized much, much greater. So.
Thank you. Really appreciate it. I'll talk to you later.
Okay, see you later.
I have a question, Kent. So now you have so many different regulations. So it seems that it takes too long to get natural enemy out. Plus, most of the candidates maybe have very low, say, parasitization rate. How do you determine if it's worth the effort? Say, what the minimum, say, success rates in order to push for releasing one specific natural enemy?
So part of the call from the I think it was 85 article and went to Canadian entomologists. At the time, Canadian entomologist was where a lot of people were putting ecology articles in entomology because they had a lot of work on forest insect pests, and a lot of the initial population dynamics studies focused on forest insect pests because we had long-term data sets. So one of their arguments was that quarantine work should start to, the data from that should start to work with modelers. Remember, this came out in the 1980s, and modelers were really getting more attention because you could have more predictive value. And so what we did is with Von Walton, we plugged in. Remember, I said that some of that data where we were looking at 20 to 60% on average parasitism on those different host plants was collected in the first generation of spotted wing. And Von Walton in Oregon said that if you plug in a reduction of 20% parasitism of the initial spotted wing population, you have a tremendous reduction by the end of the season. And so, our hope is to, when we know that the cherry grower is still going to be spraying an insecticide, but the hope is that we're going to have such an impact by the end of the season in the non-crop areas that there's not going to be pest pressure like we're seeing today going into the cash crop. And so the insecticides will not have to be used as frequently, and that they'll be more efficacious because there will be a smaller population, so less chance for resistance development because less individuals getting the pesticide pressure. So yes, we are modeling this out, and we've got now a great chance to see what happens in Canada with Ganaspis.
Okay. So if one, say, candidate has not very narrow, but can parasitize several insects, and all of them are pests, would you still consider them as a candidate, even though they have several different hosts?
So excellent question, because Trisulcus japonicus for brown marmorated stink bug attacks other stink bugs, and most stink bugs are pests, but it attacks native stink bugs. And I work closely with the person preparing that petition, and it's a good question that I maybe I shouldn't go on record saying it. My feeling right now is that if what you're releasing attacks anything that's a native, it probably should not be released.
I see. Okay.
So Kent, this is George. I'd like to follow up on that since I'm one of the people that are working with the adventive population of TJ. Should we actually be then going out there and trying to build up TJ populations with the adventive? After what we've seen in quarantine? I'm just asking for your opinion.
Yeah, my opinion is that it's there. So augmentation, and this goes back to the conversation in Spain I had, augmentation is not going to cause that much damage. All you're really doing is speeding up the process that would normally take decades for TJ to spread and see what its impact is. You're not going to take it back now. It's here. I think that the petition being looked at right now are for different populations of TJ. And so you'd have to consider whether or not the populations in quarantine have some biological aspect that make them better targeted against brown marmorated stink bug than any of the native stink bugs. And if that's the case, you may want to argue for releasing it in hopes that they interbreed or displace the adventive populations. But what you're talking about is augmentation.
Right.
Augmentation, I don't consider to have any long-term harm because it's not changing what's going to happen anyway.
Well, but if I may.
And this is Anne.
Sorry, Anne, go. Yeah, well, I just
I just have a follow-up question.
Go ahead, go.
Question, which we can talk about later too when I meet with you. But so, is anybody doing the work on comparing the the adventive populations of TJ versus the ones in quarantine? Just look at different fitness or host specificity ranges.
I assume so. I'm not part of that project, but it's a very good lab doing the work, Dr. Kim Homer. So I assume he is. I know he's working closely with the taxonomists that are looking at the genetic fingerprinting of the current populations and the populations they've got in quarantine. So I assume they are.
I mean, even within New Jersey, we have variation between our populations genetically, just looking at CO1 gene. But I don't know if Kim is actually doing any of that work. If he is, I haven't heard him talk about it, but it's an interesting avenue to go down.
I'll ask him on Monday because we're going to meet and talk about your and Tracy's objective for BMSP.
Yeah. Yeah, we'll have lots to talk about later this afternoon.
Yeah, but it's the obvious follow-up on your presentation about the importance of population level variability. So just because they have the same name, you can basically just follow up on the argument and argue that it's not really augmentation. You're introducing a new population that could have very different characteristics. So
yeah, that's exactly what happened with, I mentioned in the talk at the very end that there were 2 petitions. What we mentioned in the first petition, we were asking for both Ganaspis and Leptopolina, and we mentioned that we had built up our population from the Chinese strain, which had some, it to us seemed exactly similar. But we also mentioned in the petition that we had done our non-target work with the South Korean strain. And so part of the first petition's failure was that we were asked to repeat all the work with the Chinese population, because that was a population that we intended to release. The major differences that we found between those 2 populations is that the South Korean population had slightly better cold hardiness. And that made sense because of where we're collecting it from. I don't think that that's a permanent biological attribute. I think that's something that would change by environmental conditions you subject them to over time.
Right.
But yeah, the USDA includes that now, is that you're releasing what you study.
Yeah. I mean, ideally, what you really want to do, and that's what we all want to do, is to actually find the specific genes that determine what this particular population is going to do, and then release in accordance to the specific genetic makeup, phenotypic makeup, which we're still very far from being able to accomplish. But that, holy grail.
Yeah.
Thank you.
Okay, I think we have to stop right now. It's already past 19 minutes. So thank you, Ken, for the excellent seminar. Students who register for class will continue to stay online. Let's have a 5 minutes break, and then we'll come back join at maybe 12:04. No, sorry, 12:14.
Okay.
Thank you.
Cheng Liu,
Yes.
have you sent out the Zoom link for the afternoon meeting?
No, I didn't send another link.
And I sent you a link for it.
You did? Okay.
Yeah.
I'll look for it again.
Yeah, look for it. If not, I can send it to you again.
Oh, if you want, yeah, okay. I'll do that. Thank you.
Okay, I'll send it to you again.
Thanks. Bye.